between the corpus callosum. When you receive your sample, you'll be able to see the cerebral cortex covering the whole surface of the brain. You'll be able to identify both hemispheres of the brain. You'll be able to notice a central portion running down the middle, which we call the longitudinal fissure. This is the gap between the two hemispheres. Going across this way, we refer to this as the central sulcus. Each of these little folds and ridges increases surface area in the brain, so you can fit a lot of matter into a very small area. The raised bumps of the brain we refer to as the gyri. The little dips and folds are the sulci. So in your sample, you'll be able to identify the left and right hemispheres. You'll be able to see the hind region of the brain, which we refer to as the cerebellum, and a portion of brainstem attached. When we look at each region of the brain, going from front to back, at the front region of the head, we have what's called the frontal lobe, and this is responsible for thinking, planning, decision-making. Moving back across the brain, we have our parietal lobe, our occipital lobe, and at the back, this cerebellum. Sometimes this is referred to as hindbrain. Our parietal lobe in this region is all about body perception. It's knowing where our body parts at different times during our movement. Close behind it is the occipital lobe, and this is important in integrating all our visual stimuli. The cerebellum at the back is for our fine motor skills, balance and posture, and being able to move our body parts in a very intricate and controlled manner. Our brainstem, however, is slightly different. Think of it as your autopilot. This is responsible for all things that you're not thinking about: breathing, swallowing, heart rate. You'll be able to dissect your sample by separating the left and right hemisphere. To do this, you just need to cut down this longitudinal fissure. When we separate these two hemispheres, you can see that there's different colors of